is what is called the Golgi apparatus. Now, from the ER, remember we have these proteins. They're already connected by the amino acids are connected by the peptide bonds. And it is dropped down into the lumen, traveled to the end of the endoplasmic reticulum. The end of the endoplasmic reticulum blisters off into what's called a vesicle. A vesicle is a pouch, and it's going to disconnect from the endoplasmic reticulum with that protein inside, just like a little blister, and it's going to move from the ER to the Golgi. Now, the Golgi has been referred to as the post office of the cell because it's going to get all of these vesicles from every ribosome on the rough ER. It's going to get every vesicle carrying a protein from the ribosomes that are in the cytoplasm. And it's going to get all those lipids as well. And it's going to receive them, and it is going to further modify them. Now, when we say further modify them, what happens is the little vesicle goes to the Golgi and it breaks open and spills the content, spills that protein into the Golgi. The Golgi's job is to further modify it so it folds it into that secondary structure, that helix or that pleat, then the tertiary structure, and then it's going to join other ones for the quaternary structure. And then when it's done, it's going to send it out the other side of the Golgi into another vesicle so that it can get where it's going. Some proteins are going to need to stay within the cell. Some proteins are going to need to go into your bloodstream so they can go to other places. If this is a hormone, such as adrenaline, and your adrenal cells are making all these, then these little vesicles are going to leave the Golgi, move up to the cell membrane, and be expelled into your bloodstream so that you get that burst of energy. So some of these proteins are going to stay. We're going to talk about some enzymes and things that have to stay. If this is a lipid that needs to repair the membrane, or if it needs to be cholesterol to stay within the cell membrane, then that particular vesicle is going to stay within the cell. So some of these are going to be exported and some of these are going to stay. Now that's just like the post office. The post office is receiving mail from all over the place. It gets it from all parts of Tennessee, and then it's going to sort it out and say, okay, this mail goes to Memphis, this mail goes to Arlington, this goes to Cordova, this is going to Germantown. So it's the Golgi's job to make sure these proteins get where they need to be. Now here is another picture of the Golgi, and it was huge. So it actually was the very first organelle that was ever observed because of its size. So these enzymes that are present in the lumen, again, the space in between these membranes is called the lumen, are going to be modified and they're going to be added to. Sometimes some protein gets added to it. Sometimes some carbohydrate gets added. So depending on what is needed is going to depend on its modification. Part of the Golgi that receives the vesicles from the ER is called cisphase. And if you look up here at the top of the Golgi, it says cisphase. So cisphase is just like the front door. This is going to be where all of the vesicles from all of the ribosomes come in. Then it'll be modified all through these membranes, and the vesicles that leave are going to leave through what's called transphase. Transphase is like the back door. So all of the vesicles coming in, they come in cisphase. They get out of their vesicle, they go through the lumen, they get modified, they get added to, subtracted from, put all together, and then they are going to leave in another vesicle. You can see down here it's called an outgoing vesicle, and they leave from the back door, which we know as transphase.